So I'm Will Pidduck. I'm in third year BDC. I transferred from health science in second year, and I'm looking to do the master's program in fifth year in biomedical discovery and commercialization. So at the beginning of the year, in terms of what my research project was actually based on, we were given transglutaminase as a target in neurodegenerative disease. And what we realized is that it kind of had some varying effects in neurodegenerative disease, and we kind of wanted to look at a more broader scope of what it could actually be applied to. And what we stumbled upon was celiac disease, and this actually appeared to be one of the best applications for transglutaminase inhibitors. And so in celiac disease right now, we have some really efficacious compounds that are actually able to dial down the inflammation, but they come with some really poor safety and tolerability side effect profiles. So things like prednisone or azathioprine might have some complications like anemia or osteoporosis, and finding compounds that are specific to celiac factors but also don't carry with them the safety and tolerability concerns of these compounds in the market right now, we think is really, really important for the future treatment of these patients. And so what we ended up coming up with was two compounds, Petrichor 060 and Petrichor 037, and these were identified by our high throughput screen as potential inhibitors to transglutaminase. And we hope that these inhibitors can not only prevent human leukocyte antigen presentation and prevent that inflammation that way, but also prevent presentation as an autoantigen in this condition, and hopefully give that bimodal way of dialing down inflammation for celiac disease patients. In the pipeline right now for celiac disease compounds in development, there's actually not a representation from transglutaminase inhibitors. So there's no compounds that are actually using that as a therapeutic target, and for that reason we think that there's a real opportunity for us to capitalize on transglutaminase as a potential inhibitor and therapeutic compound for celiac disease patients. So I think it's a huge step forward in celiac disease treatment. Right now there's a small component of celiac patients that don't really respond to a gluten-free diet and don't really have therapeutic alternatives to turn to in the market right now. They could use these really strong immunocompromising medications, but you can't really use that long term with the significant safety and tolerability profiles that they have. So putting drugs like this onto the market that are actually targeting celiac specific factors might allow these patients to have maintenance therapies that can prevent this inflammation of celiac disease in the long term, as opposed to a short term acute pharmacological treatment that's on the market right now. It's probably one of the most important things in the university career and in your career as a whole, is just to see that something you're doing is applicable to the larger audience. And knowing that when you go home at night, you actually did something that's going to contribute to the welfare of somebody's life in the future. And I think being able to be part of such an important development in the celiac disease space, and kind of one of the first steps towards finding novel inhibitors for this disease and to transglutaminase as a therapeutic target, has been really meaningful to me, and it's shown that I actually could have a potential impact on these celiac patients' lives just in third year university, which is an experience that I don't think a lot of people could get.